My name is Ashanti Wright, and today our discussion is about bacterial meningitis. Meningitis is the inflammation of the membranes, called the meninges, and the meninges surrounds and protects the brain and the spinal cord. So, our most common causes of the disease is Streptococcus pneumoniae, Haemophilus influenza, some Listeria species, and the most common one being Neisseria meningitidis, or N. meningitis for short. So, three of these pathogens actually normally live in the nose and throat of healthy children and adults. However, it doesn't cause infection. The first outbreak was recorded in 1840 in Africa. However, it wasn't until 1887 when an Austrian bacteriologist discovered that the cause of the disease is Neisseria meningitidis. Where is this disease occurring, and where can we see the pathogen for the most part? Over 1.2 million cases of bacterial meningitis are estimated to occur worldwide. Although it is worldwide, the highest incidence occurs in the meningitis belt of sub-Saharan Africa. This belt's made up of 26 countries in Africa, stretching from Senegal to Ethiopia. These large-scale epidemics can occur every 5 to 12 years, and cases can reach up to rates of 1,000 per 100,000 inhabitants. And in the United States, though, which is a big difference, is that we see less than one case per 100,000 per year, with the highest infection rates in February through March. There are different subgroups or serotypes of Neisseria meningitis, and in some areas, more serogroups are present than others. This is just how they survive in these areas. About our pathogen and getting to know this guy. Humans are the only natural hosts to carry N. meningitis. It's transmitted through respiratory droplets or direct contact with these types of secretions, as well as close and prolonged contact, such as kissing, but sneezing and coughing or living in close quarters with someone can also facilitate the spread of the disease as well as the pathogen. Because remember, that pathogen can live on you or within you, but not cause disease. So there are multiple ways that we identify the pathogen. A lot of the tests that we see here that it's positive for or negative for, we've done in a class as well. It's gram positive, non-motile, oxidase positive, catalase positive, and it grows best under aerobic conditions, which makes sense if it can live in the nasal passages. It has access to air. So the pathogen can present itself in two different ways: via blood infection or infection of the spinal cord and brain, which is the one that we're talking about today, is meningitis. These symptoms can overlap, so really you won't know what the biggest concern here is until you either take a CSF sample or a blood sample, which are both ways that we can sample for the pathogens. Of N. meningitis sounds very familiar to a lot other bacteria that we've talked about in class as well. It does have this outer membrane, which is not only a way that we can tell what serotype it is, but it's also another way for it to evade our immune system. So this outer membrane protects it against phagocytosis and the complement system. We've talked about LPS in class as well, where this bacteria, its outer membrane contains LOS, lipoolgosaccharide, and this also acts as an endotoxin and is responsible for septic shock, which makes a lot of sense. And when you get a blood infection, it can be a very systemic reaction. There are many virulence factors that help it gain access to our cells. We have our capsule that helps survive the bloodstream. We have pili that are good for, or is considered a primary adhesion factor. Other types of proteins are called opacity proteins, and they help facilitate adhesion and invasion via the receptors found on the human epithelial cells, endothelial cells, and they help invade our basically our cellular system. Um, so there are ways that it can get into our central nervous system. It can do this paracellularly, passing right on through. It can also use the Trojan horse mechanism where it hitches a ride on macrophages and then is able to cross that blood brain barrier. There's also activation of signal transduction pathways that can also cause actin rearrangements, and this can also aid in pericellular transportation. There are also many genetic mechanisms that it has to avoid the immune system. Um, for example, it can mimic the immune response and basically help survive them. So, what I see that puts the disease on the map for the most part is how it's prevalent in that area of the meningitis belt of Africa. So, that is really what brings it to light for the most part. However, there was an interesting case found back in 2020 where a 5-month-old boy came to the emergency room after 6 days of not feeling too well, and they did do a cerebral spinal. They took a cerebral spinal fluid sample, and it did contain N meningitis. The lab director there, he prefers to run beta lactamase tests on N meningitis because he did hear of increasing resistance to antibiotics, and he had not observed this, but he had read literature that this is possible. So lo and behold, surprising, the rapid test um, was able to identify a beta lactamase in the isolate in meningitis. So after reporting this, there was a larger study done, and there was an agency that identified 33 beta lactamase producing and meningitis isolates from 12 other states. So this is a lot. It's becoming a lot more prevalent than people thought it would have become. And what does this mean? Well, antibiotics is how you treat a bacterial infection, and so this changes how we would do things. So usual methods would be penicillin, ampicillin, ciprofloxacin, levofloxacin, and actually after whole genome sequencing, they found resistance to all of these, which are all used for this. So they would have to either increase dosaging for this purpose and critique that, as well as maybe do a different method of trying to rid of the infection. As I previously just talked about, when there was that case study on diagnosing the disease, the most common is a lumbar puncture. That'll let us know if the pathogen did cross that blood-brain barrier. Um, Now, you also could do blood tests to see if the pathogen is in the bloodstream because it can also present itself there. CT scans are normal. They can kind of see the levels of inflammation of the meninges or if any hemorrhaging has occurred or any other things like that they can look at to suspect that it is Neisseria. So the gold standard is by culture. Um, However, PCR is useful for detecting clinical samples of a particular patient that was treated with antibiotics prior to the specimen collection. Now, after you've confirmed that it is N meningitis, you can further see what group it belongs to. The serogroup, what we said before, this is done through agglutination or real-time PCR. And to take measures even further, if you're seeing more cases in the area, next generation sequencing, whole genome sequencing, can help us identify if there's maybe an outbreak happening with the disease. I did mention treatments for the most part are antibiotics. The two most common ones in the go-tos, usually after penicillin's been given and it doesn't work, is ceftriaxone and chloramphenicol. Those are very hard to pronounce. I work at a pharmacy and still can't say medication names. Ceftriaxone belongs to a class of drugs known as cephalosporin antibiotics, and it stops the growth. They both stop the growth of the bacteria. So, which interestingly, I didn't know, Chloramphenicol can take the place of penicillin where resources are low, and it's one dose of that one is just as effective as a 5 day regime of penicillin. However, even with antibiotic treatment, 10 to 15 people in 100 out of 100 that are totally infected usually die, and up to 1 in 5 survivors will have long-term disabilities such as loss of limbs because it can cause gangrene, deafness, nervous system problems, or brain damage. What I didn't know after doing this research as well is that it was suspected that Helen Keller was suffering from meningitis. A lot of her symptoms that she wrote about and that about her how she recounts everything um, pointed towards meningitis and its symptoms, but they're just not sure if it was viral or bacterial. However, it did align with bacterial meningitis. And then she did, of course, have deafness and blindness as an effect. So there are prevention methods, and the biggest one is vaccination. We've pushed this quite a lot during COVID as well. It's because of the outlook of actually getting the disease and dealing with those symptoms and the aftermath, vaccines are your best benefit. So currently there are 3 types. There's the polysaccharide vaccine. This is usually used in outbreak response and mainly in Africa. However, the main one in Africa right now is MenACWY, and this is a conjugate vaccine. This is also used for prevention and outbreaks. And this vaccinates against the 4 serogroups that we talked about earlier as well. So this will give us the most robust immune response to the polysaccharide or to that capsule. As of right now, in Africa at least, all teenagers around the age of 14 are offered the vaccine as a part of routine immunization. In the United States, serogroup B is the most prevalent, and there is a vaccine for that one as well that's offered. So, to conclude, bacterial meningitis is a bacterial infection commonly caused by Neisseria meningitidis. However, it can have other um, bacterias that can cause the disease, but those are just not as prevalent as most others. Um, the primary ways to prevent the disease is vaccination. Primary treatments include antibiotics, but after having that disease, there can be detrimental effects to the body and to the brain, which is very important to protect, in my opinion. Thank you for listening today, and I appreciate your time. You have a great day.